Hello and welcome everyone to the YouTube channel Biology Explained. Today we are going to solve the question paper asked in the examination of Common PG Entrance Test of the subject Zoology of the year 2023. So without any further delay, let's start solving. So question number 41. Amylopsin acts upon. So amylopsin is what? Amylopsin is an enzyme, and particularly other name of amylopsin is your pancreatic amylase. Pancreatic amylase. Okay. So amylase means it is a starch digesting molecule. So amylopsin acts upon, the options are polysaccharides. Yes, it can be polysaccharide in any medium. No, this is wrong. We know all enzymes need a particular pH and temperature to act upon. So on any medium it is not possible. Polysaccharides on acidic medium not possible, because we know starch is digested in slightly alkaline medium like our mouth. There also starch digestion occurs. Polypeptide, no, this is not a protein in any medium. Here polysaccharides in alkaline medium. So option number D is the correct answer. Question number 42. Carbon dioxide is carried in the blood in the form of sodium carbonate, potassium carbonate, sodium bicarbonate, magnesium bicarbonate. Right answer will be option B, that is sodium bicarbonate. Question number 43. Reabsorption of glucose from glomerular filtrate in the kidney tubule is carried out by reabsorption of glucose. So glucose is carried by our active transport. Option number C is the right answer. Question number 44. The number of internodal pathways extending from SA node to AV node is internodal pathway. So answer is actually 3. Let's understand this with the help of this diagram. Here you can see, here our SA node is present and here our AV node is present, and the number of internodal pathways is 1, 2 and 3. So 3 pathways are there. So our answer is option number B, that is 3. Next, moving to question number 45. Which of the following is an incorrect statement about metabolism? So options are: option A, metabolic pathways are always irreversible. Option B, each metabolic pathway has first step committed. Option C, metabolic pathway follow only oxidative process. Option D, metabolic pathways are regulated. So when we look at these options, I find most of the options incorrect here, but the question is asking an incorrect. Only one incorrect option you have to follow. Let's decipher each and every option. The first option is metabolic pathways are irreversible. It is not necessary that metabolic pathways will be irreversible always. It can be reversible also. Okay, so this statement is incorrect. But let's look at other statements as well. Each metabolic pathway has first step committed. This is also not necessary that first step will be always, first step will be committed step. So this is also a kind of incorrect statement. Metabolic pathway follow only oxidative process. Only. The word only makes it totally incorrect. Metabolic pathway need not to follow only oxidative process. It also can follow up reductive process. Metabolism means there can be oxidation, there can be reduction, there can be anabolism, there can be catabolism. Metabolic pathways are always regulated. So this statement is absolutely correct. Metabolic pathways are always highly regulated. So the most incorrect statement from these options will be option number C. Otherwise, rest two options are also incorrect, but the word only here makes it the most incorrect statement. Therefore, the answer will be option number C, that is metabolic pathway follow only oxidative process. Next, which of the following is not a substrate for gluconeogenesis? Gluconeogenesis means synthesis of new glucose molecule from non-carbohydrate substrates. So options are lactate. Yes, lactate is a source of substrate for gluconeogenesis. Glycerol, yes. Alanine, yes. It is an amino acid. This is also a source for gluconeogenesis. So the answer, question is asking not a substrate. So option number D, that is acetyl coenzyme A, will be the right answer. Here you can see, you can take a screenshot of this. I have listed out the substrate. Here glycerol can be a substrate for gluconeogenesis. Pyruvate, of course, it is made from amino acids, lactate, and alanine and other amino acids also contribute to the formation of pyruvate. And from pyruvate, glucose can be synthesized through the pathway of gluconeogenesis. Next, moving on to next question. Question number 47. The net gain of ATP through beta oxidation of palmitic acid is, okay, palmitic acid is 16 carbon chain long. So we are going to have 7 cycles of beta oxidation. So to summarize, what will happen? In each cycle, one FADH molecule is released and one NADH molecule is released. So in 7 cycles, how many FADH? 7 FADH2 and 7 NADH2. So what we are getting, each FADH, one FADH is equal to 2 ATP. So therefore total number of ATP is 7 into 2, that is 14 ATP. Similarly 7 NADH molecule means 1 NADH molecule values is equal to 3 ATP. So 7 NADH molecule is equal to 7 into 3, that is total 29 ATP. And what else we are getting through beta oxidation? 8 molecules of acetyl coenzyme A. We know that one molecule of acetyl coenzyme A, when enters into our Krebs cycle, it yields 12 molecule of ATP. So when we are getting 8 molecules of acetyl coenzyme A through beta oxidation, how many molecules of ATP will be generated? 8 into 12. That is equal to 96 ATP. When we are totaling it, we are getting 131 ATP. But this is not the final answer. The question is asking the net gain of ATP. Net gain means what amount of ATP is utilized for beta oxidation. For activation of fatty acid molecule, 2 molecule of ATP is utilized. So what will be the net amount? 131 minus 2, that is equal to 129 ATP. So the right answer for this will be option number C, that is 129 ATP. Next, question number 48. Which of the following enzymes is not involved in urea cycle? If you remember the urea cycle, then this is the easiest question. Carbamyl phosphate synthase, yes, it is present in urea cycle. What else? Arginosuccinate lyase, yes, this is also present. Arginosuccinate synthase, this is also present. Succinate dehydrogenase, no, this is an enzyme present in Krebs cycle. So the question is asking not involved. So option number D will be the right answer. That is succinate dehydrogenase enzyme is not present. Next, question number 49. Which of the following is classical uncoupler of electron transport chain? Options are antimycin A. This is an example of inhibitor molecule. So this is not the answer. Cyanide, this is also an inhibitor. This is not the answer. Thermogenin. Yes, thermogenin is an example of uncoupler molecule. So option number B, that is thermogenin, is the right answer. Question number 50. Which of the following makes eukaryotic DNA replication different from prokaryotic DNA replication? Options are use of DNA primer rather than RNA primer. No. In eukaryote as well as prokaryotic DNA replication, RNA is only used as primer. So this option is totally incorrect. Use of different enzymes for synthesis of lagging and leading strands. Yes, this can be true because there is a slight difference in the type of enzymes which is used in the DNA replication of prokaryotes and eukaryotes. So next, moving to next question. Question number 51. Which of the following RNA polymerase targets only mRNA genes during transcription? mRNA genes, remember the code word RMT1, 2, 3. That is, rRNA is transcribed by RNA polymerase 1, mRNA by RNA polymerase 2, tRNA by RNA polymerase 3. So, according to this code, our answer will be for mRNA, what we are getting? Option number B, that is RNA polymerase 2. Next, moving on to next question. Which of the following complexes is generated with the release of SN-RNP U4 and the formation of a lariat in the spliceosomes during the splicing of pre-mRNA in eukaryotic nuclei? The answer will be, that is complex C1 complex. Here you can see this with this with this diagram. You can take a screenshot if you want. So this is the complex C. After U4 is released, what we are getting? This complex C1 we are getting. Next, question number 53. Lac operon of E. coli exhibits, okay, a repressible negative control transcription. No, it is an inducible. So here is option number B, inducible negative control of transcription. Yes, option B is the right answer. That is, it is inducible negative control of transcription. Next, question number 54. Which of the following is not sex-linked trait in humans? Not a sex-linked trait. Okay. Duchenne muscular dystrophy, it is X-linked. This is a sex-linked trait. That is why this will be not the answer. Color blindness, of course, this is sex-linked. Again, X-linked. Hemophilia, this is also a sex linked, so this is not the answer. Baldness in males, yes, this will be the answer. This is not sex linked, rather this is sex what influence trait. This is sex influence trait. Next, question number 55. In mutational event, when adenine is replaced by guanine, it is a case of what? When adenine is what? Adenine is a purine. It is replaced by what? Guanine. Guanine is also a purine. So when purine is replaced by purine, this type of mutation is called transition mutation. Remember this diagram, students. When a adenine or a purine is replaced by a purine, we are calling it transition. When a pyrimidine, that is, see this cytosine, is replaced by another pyrimidine, this is also transition mutation. Means purine if replaced by purine, or pyrimidine if replaced by pyrimidine. This is example of transition. But the opposite if happens, means what? If a purine is replaced by pyrimidine, or a pyrimidine is replaced by purine, this is a case of transversion mutation. Okay, remember this, students. So option number B will be the right answer for this question. Next, question number 56. When a transposon moves out of donor side, what is transposon? Transposon is your jumping genes. It is a jumping gene. Okay. Transposon moves out of donor site and inserts into new site, leaving a double-stranded break. It is leaving a double-stranded break at the original site of the donor DNA, which may not be repaired. Okay, which may not be repaired again. Then it is called which type? It is called conservative transposon. In case of replicative, it will be repair. Replicative and conservative are same. But in case of replicative, it is repaired in the original site. But the question is asking may not be repaired. If it is written may not be repaired, answer will be option number B, that is conservative transposition. Question number 57. Which of the following is not a criterion of cytoplasmic inheritance? Options are A, lack of chromosomal location, B, association with parasite symbiotes or virus, C, reciprocal differences, D, somatic segregation. So, if you look at these options, right answer for this will be option number B, that is association with parasite or symbiote. This is not a criteria for cytoplasmic inheritance. Next, question number 58. Fate map of embryo is prepared in which stage? Answer will be option number C, that is gastrula stage. 59. Which of the following is associated with centrolecithal eggs? Equal holoblastic cleavage? No. Discoidal meroblastic cleavage? No. Superficial meroblastic cleavage? Yes. Option number C is right. Remember, students, the rate of cleavage is always inversely proportional to the amount of yolk present. Wherever the yolk will be present, cleavage will not occur there. In centrolecithal egg, suppose this is the egg, yolk is present here in the center. So how the cleavage will occur? Cleavage will occur here. Only superficially the cleavage will occur. That is why option number C is the right answer. Remember the organisms on which these types of cleavage are occurring. They can also be questions in the upcoming examinations. So, for example, superficial cleavage, it occurs in case of insects mostly. Next, question number 60. Which of the following exhibits epimorphic regeneration? Simple epimorphic regeneration occurs in case of salamander. You can see here whenever the limb of the salamander is amputated or cut, what happens, this regeneration occurs here itself. This type of regeneration is called epimorphic regeneration. Next, question number 61. The RNA world hypothesis to explain origin of life on Earth was proposed by, answer will be option number C, that is Walter Gilbert. Next, question number 62. The first three-toed fossil of horse was, remember first three-toed, remember this timeline of the horse evolution. It is asking first three-toed. Okay, Pleohippus is just primitive to the modern horse, so they were having single toed. Okay, Orohippus, this is ancient one, they were having four toes. Okay, Meryhippus, they were also having what? How many toes? One toe. Only Miohippus here is the option which is having three toes. So option number B will be the right answer. Here you can see Miohippus is here, it is present. But if the option would have given Mesohippus, then it would have been the more correct answer for this. Mesohippus and Miohippus both were having three toed. But since none other of these options contain any other organisms like Mesohippus, therefore we are considering Miohippus to be the right answer here. Next question, question number 63. Which of the following occurs when geographic isolation creates a reproductive barrier? Very simple question. Answer for this will be allopatric speciation. Question number 64. The earliest fossil of prehistoric man is, okay, earliest fossil. Again, options are Dryopithecus, Sivapithecus, Ramapithecus, Australopithecus. Right answer for this will be option number A, that is Dryopithecus. If you are facing any difficulty to remember this, I am giving you a mnemonics to remember. Here you can see I used to remember this with the help of this mnemonic. Please do remember, apple has essential nutrient for humans. Here you can see Parapithecus, do for Dryopithecus, R for, remember, Ramapithecus, A for Australopithecus, H for Homo habilis, E for Erectus, N for Neanderthalis, F for Fossilis, and H for Homo sapiens. This mnemonics, remember this mnemonics, and most of the evolutionary question you will be easily able to answer. Next, question number 65. Crypsis is a type of, which type of behavior? Crypsis is a type of defensive behavior. What happens in crypsis? The organisms mimics the surrounding and it tries to hide itself so that it can avoid itself from the predators. Next, question number 66. The sleep-wake cycle is an example of, this is an example of circadian rhythm. Next, question number 67. Which of the following cloning vectors can carry the largest insert of foreign DNA? Largest insert of foreign DNA. Option are plasmid. No, they carry a very small. Lambda bacteriophages. Okay, they carry larger but not largest. Cosmid, of course, they are again larger. But here is the last option, that is East artificial chromosome. So option number D will be the right answer, that is East artificial chromosome carries the largest insert of foreign DNA. Next, moving on to next question. Question number 68. Which of the following is used for detection and analysis of proteins? Okay, options are southern blotting. Southern blotting is used for DNA. Northern blotting is again used for RNA. Western blotting is used for detection of protein. Sorry, it is used for detection of proteins. But the question is asking detection and analysis. Okay, detection and analysis. Here is another option, that is Eastern blotting. So option number D, that is Eastern blotting, will be the right answer. That Eastern blotting actually helps in detection as well as post-translational modification is also detected with the help of Eastern blotting. So Eastern blotting will be the right answer. Next, question number 69. Which of the following is not a technique used for detection of genetic disorders? Not a technique. Again, option number A, that is PCR, polymerase chain reaction. This is a very essential thing for any genetic practice. So this cannot be, this is of course in use. Then DNA sequencing, this is also an essential practice of molecular biology. This is also in use. Karyotyping, yes, of course. Next, last option is ectogenesis. Ectogenesis is actually related to the concept of artificial womb. Okay, artificial womb. This is something related to reproductive biology. Here I can show you here how the embryo is developed artificially in the laboratory. This ectogenesis talks about it. So option number D will be the right answer. It is not a technique used for detection of genetic disorders. Question number 70. A typical synaptic cleft is. Okay, this is a fact based question. The right answer for this will be option number D, that is 20 nanometer wide. Question number 71. Which part of some nerve cells is myelinated? This is a simple question. We know the parts of nerve cells are dendrite, axon, and cell body. So dendrite is not myelinated, neither cell body is myelinated. Myelinated portion is your axon. So answer for this will be option number B, that is axon. Question number 72. The nature of conduction of nerve impulse is. The answer for this will be option number C, that is electrochemical. Next, question number 73. Which of the following is not an inhibitory neurotransmitter? Okay, this is asking about inhibitory neurotransmitter, not an inhibitory. Okay, first option is gamma aminobutyric acid. This is nothing but your GABA, what we call as GABA. And GABA, you know, is an inhibitory neurotransmitter. Glycine is also inhibitory and serotonin is also inhibitory in action. The only option left is option number D, that is glutamine. That is not an inhibitory neurotransmitter. Here you can take a screenshot of this. Inhibitory neurotransmitter is glycine, GABA and serotonin, whereas excitatory are glutamate, aspartate nitric acid, and here it is both. Next, question number 74. Binding of a signaling molecule to which type of receptors leads directly to change in distribution of ions on opposite side of the membrane? Right answer for this will be option number C, that is ligand gated ion channel. Question number 75. A decrease in level of estrogen and progesterone causes. Options are option number A, loss of endometrium. Yes, this is right. Remember students, progesterone is also called pregnancy maintaining hormone. Pregnancy maintaining hormone. Means what? This progesterone ensures that the layer of endometrium is intact in the uterus. Suppose this is the endometrium layer, this layer should be intact whenever the amount of progesterone is high. Whenever the level of progesterone decreases, what happens? This endometrium layer is shed off, and what happens? Menstrual cycle again enters into the bleeding phase. So here it is the question is decrease in the level of estrogen and progesterone. The option A is right, that is loss of endometrium. But here in this question a twist is there, that is option number D. If you see, constriction of uterine blood vessels leading to slowing of uterine epithelium. This answer is also correct because both the questions have the similar meaning. So both these options are actually correct. But since loss of endometrium directly here it is written, so I am considering option number A to be the right answer. Next, question number 76. Which of the following proteins is not present in cell junctions? The type of proteins which are present in cell junctions are cadherins, yes. Selectins is also present. Integrins is also present. Transferrin is not present in cell junction. Transferrin, as the name suggests, it helps in transfer of molecules. So answer for this will be option number D, that is transferrin is not present in case of cell junction. Next, moving on to next question. Question number 77. The specificity of an antibody is due to the specificity. Always remember the structure of antibody, and this specificity to the antibody is provided due to this variable region present in the antibody structure. So options are its valence, no. The heavy chains, no. Not only heavy chain, light chains are also responsible. The FC portion is this portion, this constant portion. This is not responsible for the variability. The variable portions present of portion of heavy and light chain is responsible for the variability. So option number D will be the right answer. Next, question number 78. Hypophysis refers to. Hypophysis is a method of induced breeding. It is a method of induced breeding in case of fishes. So option are molting, no. Eye stalk, ablation, no. Hybridization, no. Option number D, that is induced breeding with pituitary extract. What happens in fishes, those which are not breeding, what happens, we take a pituitary extract and we inject this pituitary extract into the fish so that this fish get mature and they start breeding. So option number D will be the right answer. Next, question number 79. Viable material of endangered species can be preserved by. Okay, we are talking about viable materials. Okay, viable materials. When we are talking about viable materials, the options are gene bank. Yes, option number A is the right answer. Gene bank is the right answer. Gene library has only DNA. Okay, herbarium contains plant materials, and gene pool is the total sum of genes, that is genome I'm talking about. So this is a photograph of a gene library. This is how gene library looks like. Different type of materials can be preserved, like any embryo, egg cells, sperm cells, or any plant part, or anything of the organisms can be kept to preserve it. Next, question number 80. Which of the following is a tropical tussar silkworm reared in India? This is again a factual question. Answer for this is option number B. That is Antheria mylitta. So this is all about the CPET 2023 question paper. Okay, this is the diagram of this Antheria mylitta. You can see. Thank you for being there and thank you for listening to the video. And I hope this video was helpful for you. If you are liking this video, kindly subscribe to my channel and support me so that I can make more such videos for you. Thank you all.